Thank you for the invitation. It's really always a pleasure to be here. It's my second time giving a talk. I was invited by the Department of Math by Mauricio Barahona quite a few years ago. And it was great, and again, it's now nice here. I mean, so you have an amazing group of people, so I'm really very happy. Thank you. Okay, so maybe I'm a bit optimistic with the word rich, because what does it mean, a rich environment? And I have to tell you that we are working towards really having a really rich environment. So what by rich I mean here, maybe non-trivial, non-static, a little bit somewhat non-one-dimensional. That's what I mean, and I will tell you in a minute. So, but anyway, I just want to lower the expectations regarding what a rich environment means. So anyway, since this is a physics of life, let me see if okay. Since this is a physics of life seminar, I'm going to assume that many of us in the audience at least have a physics background or a physics spirit. And I don't know about you, but when I moved to systems biology, coming from a background on nonlinear and stochastic dynamics, I was fascinated by molecules, by molecular biology, that the fact that you can have a lot of dynamics and noise at the level of molecules. But then little by little, I became more interested on the thing that is a little bit higher in the scale, which is cells. Because in some sense, maybe I'm going to throw the word here of agency. So you can say that cells have somewhat of an agency, if you will, whatever. I'm not going to enter into the philosophy, but cells use this molecular repertoire of processes to do things, and one of the things that the cells do is to really respond to environmental conditions. And environmental conditions is not only whatever externally we apply to the cells, but also maybe their local environment. These are identical cells, and yet their local environment is slightly different, and this environment is definitely rich. So ideally, we would like to understand how to correlate the local environment and the global environment of a specific cell to whatever it does. Now, this is really rich, but definitely we are not there yet, so I'm going to tell you what we have been doing in that regard. Anyway, so this is basically a restatement of what I just told you. So you can have cells here, and the cells are receiving many kinds of different inputs, not necessarily chemical, they can also be mechanical. We have been talking with some of you about this. And the cell has to integrate all these inputs, so it's a problem of integration, but at the same time it's a problem of taking into account the fact that these inputs change in time in non-predictable manners sometimes, right? So, so the question is, how do cells respond to this type of situation? Can we understand this? Maybe we can frame this in terms of adaptation, or maybe high-level adaptation, which is what I would like to frame it in terms of. But what I would like to say from the very beginning, just to frame the content of the talk, is that this is a very similar situation to the one that brains face, no? Brains have also a multitude of different kinds of signals. They have to integrate all the senses, and these signals are time-dependent, so definitely this is a complex environment. Of course, people have been trying to understand how brains work in these rich environments, and there, of course, we know the structural properties, we know the mechanistic properties of brains, which is that we have huge numbers of neurons connected in a very complicated manner. So we know what the brain is made of, and the next question is, okay, so how does this thing give rise to the complicated responses that brains have to a rich environment? And of course, there is, as you know, better than I, there's a huge gap. There's a huge gap into our knowledge of what happens at the cellular level in the different neurons and in the networks with what happens to human beings and how do human beings respond. And for that, of course, I can see Mauricio there, sorry. So how do human beings respond? And of course, this is a topic of cognitive science. And in cognitive science, this has been addressed for, I would say, a century, right? So in cognitive science, and this is maybe one of these paradigms that have emerged at the highest level, which is that, look, we have a system, could be a brain, and it has a complex environment, and this paradigm, which is not ours, is basically that this brain is encoding this environment in an internal representation, and this internal representation guides the behavior of the system, okay? So, and this is framed in terms of learning, and this is ideas that it's not mine. They have come with multiple conversations with my colleagues, Aneta Koseska, Jeremy Gunawardena, and Dietmar Schmucker. We are sharing a project in dealing with these things right now. Now, so this means that this is, of course, a very high-level abstract interpretation of what's going on, but this allows us to, or this allows, in principle, for instance, at the level of psychology and cognitive science, to establish phenomena such as habituation, sensitization, or conditioning that we know that the animals are following. And there is some evidence that cells, I'm not going to enter into this today because I'm going to deal with something very different, but just to frame, there is some evidence that the same thing is happening at cellular level, and we are interested in pursuing this. Okay, and I really recommend this review by Jeremy, he published 4 years ago already, but it's very nice. So anyway, this doesn't solve the gap. There is still a gap. And the gap is that on the one hand, at the bottom, we have the neurons, and on the top, we have our brains, which have behaved in this manner. And then the question will be, okay, so these internal representations, how are they decoded? I mean, to be honest with you, I mean, this kind of approach is not very satisfactory until I really want to go down and find the mechanisms. So I guess we are all more or less in agreement in this room that the idea would be to find the molecular basis of these internal representations. Because so far in cognitive science, this was not very much studied because you had to go down to a brain which is composed to 10 to the 10 neurons with huge, complicated networks. So we cannot do it. It's not so easy to do it in brains. So our approach is that, well, maybe brains are too difficult, but maybe cells are not so difficult. We know, as I was telling you, and I haven't shown you the data, but there is already evidence that cells implement this kind of learning strategies. I mean, there is a clear analogy between a network of neurons and a network of genes and proteins. So this analogy is there. So maybe we can consider cells as a model system in which we can maybe try to address this and connect this gap to do this. So in the case of cells, the environment could be mechanical and biochemical signals, if you will, electrochemical or whatever. And then the behavior would be motility, differentiation, all kinds of decisions that the cells are making. So, of course, both the signals and the behavior are much simpler than in brains. So we would like to follow in this direction to maybe help us have maybe cell biology in that regard can help cognitive science and vice versa. That should be the idea. So now instead of a brain or a box, we have a cell, and the cell contains multiple networks, and these networks are, of course, there is an elephant in this room, which is neural networks, artificial neural networks, machine learning, and we know a lot now, after so many years, about different architectures that are amazing at processing information. And one of the things that we are doing in our group that I'm not going to be telling you about today is, well, how do these networks are able to implement these things? And we want to work in these directions, but for that, we need to be able to apply complex, really rich environments to the cells. But we are not there yet, okay? So what we have done at most is maybe we have already recently discussed and tried to come up with molecular mechanisms that explain phenomena such as, for instance, habituation. This is something that we did a few years ago, again, with Jeremy Gunawardena and another colleague, Rosa Martinez-Corral. And here you can see the habituation characteristics. I'm not going to go again into the detail. You have multiple presentations of the same stimuli, and the response goes down. This is the opposite of sensitization. And from a purely computational, theoretical, if you will, level, we saw that a relatively simple network of two couple feed-forward loops, which are based on at the level of post-translational interactions, can explain this habituation phenomena with all the different hallmarks that the habituation follows. These hallmarks were defined by cognitive scientists many years ago. But again, we have to validate this. And the problem at the time of validating is that there are two problems. Again, the lack of right now of our ability. I'm not saying that some of you may be working with very sophisticated microfluidic systems that are already allowing you to do this, but we don't have it yet. But also the other problem is that, well, to encode the internal representation, the state of the cell is already there, and usually cells, living cells that are doing stuff, have a very complicated internal state, because they are storing the history of a phenomena. They are trying to survive, so they are doing stuff. They are dividing or they are fighting, whatever. So the internal state of these cells is very different, so very difficult. So the question is whether we can find living forms with internal states that are simple enough. Because if they are very simple, then maybe I can try to identify the internal representations, okay? Now, two things. The first one is that now I'm going to switch. So this is the question. But now I'm not going to be talking about these very broad things anymore. I'm going to be talking about something very specific. And I just decided to tell you a little bit of a collection of things that we have been working on a system in which the internal states are relatively simple. I'm going to tell you already what this system is. And I'm going to start talking about bacterial spores. Okay? So a part of my talk, maybe all of it, but I'm not sure. It depends on how we go. Is going to be devoted to these organisms. So these are living systems that are very simple because they are dormant. They are really dormant. So these are not like bacterial cells in a stationary state. These are not like differentiated cells in mammalian systems. These are not like persistent cells in persistent bacteria. These are really dormant, okay? They have almost no water. They have almost no ATP. They cannot spare ATP, so their internal state is as simple as it can be. This is as close to non-dead as you can imagine, but they are alive, which I find it very interesting. So now I would like to tell you a little bit about things that we have working, going away from, for the moment, explicitly about this learning idea that I just shared with you as a plan of what we want to do in the future, but I want to tell you what we have done in the recent past with this system. And I have to stop now. The second thing I wanted to tell you is that now I have to thank my collaborators. So I'm putting my thanking slide in the middle of the talk because I cannot do otherwise. I'm not going to show you experiments from our lab. I'm going to show you experiments from the lab of Grosswell at UCSD. This is us. As you can see, we have been working together for many years because I was very much younger. I'm older now, so you can see that this is a very old picture. So we have been working at this for many years. It's still the same. He doesn't age, but I am like this, older. So we have been working together for 20 years, basically. And I just want to tell you some things that we have been doing. He has these amazing experimental setups. And then we have all this data. And then a physicist comes and tries to have fun with whatever they are observing that's there. So this is the people, and I'm going to be naming them as we go. But I just wanted to make the thanks right now. So let me go back to the system. These cells, these spores are metabolically dormant, and this is an understatement. They are really dormant. They can be in this state for thousands of years. Okay, so this is not thousands of years. You take them out of the rock. You give them the right food, and as you will see in a minute, you will see right now, in 10 minutes they wake up. Okay, so it's in my opinion, this is an amazing system. Now let me show you first how they form. Okay, so you have here a population of, this is Bacillus satalis bacteria. Green means that they're happy. You will see some red, which means that they're not so happy. They're doing something else. Let's not forget about this. But the important thing is that you will see white things appear, and these are bacterial spores. When the white little things appear, this means that the cells have lost almost all of the water. This is phase contrast image. And then white means that there's basically way less water than they were when they were black or green or red. Okay, so you see here a bunch of these spores. We are going to show them as little black things, which means that they don't have a lot of water. So how do we wake them up? So one way of waking them up, this is not like bacterial seeds. Sorry, plant seeds. These are not plant seeds. You take a plant seed, you put it in water, and then it comes out. You take spores, you put them in water, and they are still there. I'm telling you, this is the way we store them in freezers for years, right? So water, nothing. But if you give them the right source of nutrient, if you will, which we call a germinant, then it's going to wake up. And the germinant that we have been using, there are multiple, not too many, but there are a few, is L-alanine. So it's the L form of the amino acid. I'll come back to the L thing. So we have been discussing earlier today about chirality. So the chirality is going to become important in a minute of this amino acid, okay? So it's L-alanine. So now if we give L-alanine starting at this point, so this is at this moment, we drop L-alanine and we keep it there for a while. These are minutes, and you see that in 10 minutes, this thing becomes black, which means that water comes in. And the next step that I'm not showing is that then once the water has come in, the bacterium start to grow. I'm not showing you the growth part, I'm just showing you the hydration part. And this happens when you add this alanine, which is a germinant, okay? But I told you that I wanted to have some kind of rich environment. And rich means that at least it's not constant. So it's a bit poor, it's a poor definition of rich, but means that it's not constant. So now instead of having continuous germinant, let's see what happens if the germinant is time dependent. That's my definition of rich. at the beginning. Okay, so one single input that is time dependent. By the way, these are, so now I show the first picture. These are experiments that were done by Kaito Kikuchi, who was a PhD student in Guro's lab at the beginning. That's a few years ago when we did this. So anyway, what happens when we, instead of having a continuous germinal, we give pulses of germinal? So periodic, again, not very rich, but at least time dependent. We physicists love periodic driving, no? So what do I need to tell you? So basically we have 3-minute pulses of L-alanine every hour or 2 hours, okay? So it's a periodic driving. Now, what happens seems to be very boring. 1 pulse, nothing happens. 2 pulse, nothing happens. These are hours, so it's every 2 hours. 3 pulse, nothing happens. And then we go back again. So it seems that nothing is happening, which might be okay. Well, I mean, I don't know. This is a concentration of L-alanine that is not enough, clearly, to induce germination in this spore, and that's why it happens. So maybe that's it. So it will be a bit boring. Yes, yes, Ron. Yes, yes, sorry. This is the flow chamber. Okay? Well, that is not enough to produce germination at high levels in this particular case. So basically, you flow it in continuously or in pulses, 3 minutes, every 2 hours. So you have your cells here, and then what you can do is you can do a statistical analysis. I was showing you just one cell, one spore. What happens when you have hundreds in this device and you flow in your periodic train of pulses of germinal, okay? Let me show you what happens. So here you have the field, and then here you have 45 spores in time, and then you will see that now this is no spore, spore, spore. Now 3, you have a pulse, 3 of them respond. You have another pulse, a lot more respond, and you have a lot more response. So you see every pulse, which happens every 2 hours, is eliciting some response. So it's not that nothing is happening. Something is happening. At the population level, there is an increase in the percentage of spores that germinate, okay? So far, so good. Well, but I'm sure that you're already perceiving something here. You're saying, something is weird, or something is especially not worthy. And what is not worth it is that I can calculate the probability of germination, or the fraction of cells that become dormant. So these are inversely related, of course, no? Fractions of cells that become dormant, or probability of germination. After the first pulse, most of the cells remain dormant. The probability of germination is low. But what happens after the second pulse? You have seen it already, I'm sure. There are much more black things, which means that now many of the cells go down. The dormant cells, half of the dormant cells now germinate, which means that the probability of germination really grows up a lot, okay? So if this was independent events and the spores were not doing anything, the probability of germination, I hope you agree with me, would be constant. But the probability of germination is not constant. The second pulse, many more germinate. So I would like to call this, at least at the population level, this is some kind of sensitization. So this is my reminder of what I told you at the very beginning. So these spores are getting sensitized at the population level in such a way that this thing goes up. So the question is, why does this happen? So clearly, the cell, the spore, must store an internal representation of the history. That's all. I'm calling it internal representation. But something is just changing. Something is just changing in the spore, in the spore's internal state. What can it be? Okay, so now there are not too many options, because these cells have nothing. These cells have no energy. They have no water. Nothing moves in here. Water will come later, and then maybe this will wake them up. But they don't have energy to spend monitoring the environment if they want to stay like this for thousands of years. So they have to do something passively. And then after some time, we came to the realization, or with this the hypothesis, that what could be happening is ion flow. Okay, ion flow is cheap because, of course, as you know, there are huge concentration gradients of different cations in between the interior and the exterior of these cells: bacteria, vegetative bacteria, neurons, mammalian cells, and spores. Okay, there are huge concentration gradients, and if I am able to open an ion channel that I could open for free, no, it's just a conformational change, so it doesn't cost me ATP. No, if I open an ion channel, then I'm going to change something, okay, in the internal state. And I mean, it's not that we came to this idea because we had seen in previous year, after this 20 years in which we have been working together on this type of things, that bacteria, not spores, but in general bacteria, and in particular things such as bacterial biofilms, really use ions and use electrochemical signals to transmit information. So this is something that we observed already more than 10 years ago, and these are potassium waves in a bacterial biofilm. I hope I will come back to bacterial biofilms in a minute. And this is, of course, just all propagation of potassium in a bucket brigade kind of way from cell to cell, and this is transmitting the information of the stress here to the outside. So basically, what this is transmitting the stress information from the center to the outside. This thing, these ions, it's not only through the communication is not only inside the biofilm. It can also happen between. It can also happen between biofilms. So the ions, of course, are going to diffuse very fast here, and then they happen to oscillate. I'm not going to enter into this. So these are two oscillators coupled through potassium exchanges, and they are synchronizing. So ions are really playing a role. We keep seeing them over and over again. We also saw them that in this was potassium so far, but even magnesium is also helping protect ribosomes against certain types of antibiotics. So we can see that ions can also promote antibiotic resistance in cells. This is something else that we saw, again, in bacteria, not in spores. But we came up already pre-conditioned to think that ions played an important role. And now we see that maybe here as well, because it's very cheap to alter the internal state of the cell, of the spore, if the internal state is the concentration of a given ion. Okay, so that's the idea. Also, and I'm not showing you data, but this is something that is in the literature. For instance, it's known that potassium concentration is higher in spores than in vegetative bacteria. Of course, people have studied, microbiologists have studied spores for ages, and they have done all the census of everything that includes a spore. And then one of the things that is known is that, for instance, the potassium ion, is higher in spores than in vegetative bacteria. Yes?
But how does it now relate when you talk about this L-alanine?
Yeah.
When you talk about L-alanine, the question is how it regulates some ion channels?
Well, I'm not entering it. I am going to make assumptions. I think I can.
And then, of course, you have the dehydrated state, right? It's not so easy to have.
Yeah. Well, okay. So I am not. We are not molecular. I don't consider definitely myself a molecular biologist. I don't think that Guro is either. So we haven't done the molecular biology, but we used knowledge that it's already there, and we know that there are receptors. There are receptors on the coats of these spores. One of them is called GER-A. This receptor, it's called GER-A because it binds to germinans. So we know that there is binding between these germinans and this receptor, and we assume that what this is causing is some kind of some kind of conformational change that is going to, in turn, make an ion channel open. Okay, if you want. And then the spore has a lot of potassium, so there is a large potassium gradient. The channel opens and potassium leaves. That's the assumption. Okay, so germinant comes, binds to the receptor. This opens the ion channel. This is all. I mean, we are not molecular biologists. We are just using things that are known. If you ask me, I think the coupling between the GER-A receptor and the ion channel, I don't know to what extent this has been well established at the level of structural biology and how the conformational, I don't think so, but this is our assumption, okay? Now, we are not doing molecular biology, but we are doing cellular biology and cell biology. And for instance, one thing that we can do is we can use potassium dyes, such as this fluorescent potassium dye, which is extracellular, and then you add the potassium dye to your cells, to your spores, and you can see what is the signal on the outside of the spore in time when you produce the pulses. And then you see that when the pulse arrives, there is a jump in the dye, which might indicate, in our opinion, that potassium is leaving. Potassium is leaving, pip, and then it relaxes in a period of with a time scale of around an hour, and then potassium leaves again. This is a control. Well, if we want a control, we can use a blocker. We can use quinine that specifically blocks this ion channel, this potassium ion channel. So if we add quinine, we block this potassium ion channel. Now potassium cannot leave, and indeed it doesn't leave. So this is we are seeing. So I mean, our assumption is that for whatever reason, a germinant comes, binds to the GER-A receptor, and then the ion channel opens and potassium leaves. In that way, then the idea would be that our internal state is it's super simple. Our internal state is a concentration of potassium. Now, now we go, a physicist comes, and then let's do some modeling, a little bit of modeling, no? So then what we can do is, look, if we are talking about ion channels that open and close, and the ion channels are gated by different kinds of things, in this particular case, the ion channel would be gated by the germinal binding to the JA receptor. So basically we can use, I mean, of course, the history of models of electrophysiology, of models of electrophysiology is, I mean, it's super dense because we have neurons, and neurons have all these channels, and the channels are gated by voltage. In our case, this channel is gated by the germinal binding. But we can build if you will, a Hodgkin-Huxley-like model, and I'm not going to go into the details. I just put here this, I mean, it makes no sense, and I don't want to spend a lot of time on this because I have many other things to tell you. But we have an equation for the membrane potential, an equation for the gating variable. This is very standard Hodgkin-Huxley. The difference now with Hodgkin and Huxley is that bacteria are very little. In Hodgkin and Huxley, we assume that a neuron is very large, and when the ions go in and out, the concentration of ions don't change. In these models, what we usually assume is that the levels of potassium are changing, okay? At least the internal one. The external one, we can assume that it's basically the area around the cell. Okay, so we have also equations for the extracellular and intercellular potassium levels, and of course we have, we have the Nernst potential of the channel, and then what this model basically is establishing or making the hypothesis that there is a germination propensity which is linked to how little potassium you have. The less potassium that you have, the more propensity to germinate. That's our assumption. Okay. So at the very beginning, you have a lot of potassium. You are not germinating, but then the less potassium you have, the higher the propensity. And this is explained by this model. And at some point, there is a threshold. And when the propensity reaches a threshold, which means that when potassium is low enough, there's going to be a firing, if you will, and then the water is going to come in, and then the rest of the things are going to happen for different other methods, okay? But the idea is that we have, everything depends on the initial potassium content and the potassium efflux. So this is established by the sporulation process. When the spore was formed, it is formed with a certain concentration of potassium, okay? That we can also perturb, and then another thing is that the efflux, which is the ability of the channel to open and let things out. And then these two things determine the dynamics of the potassium content, and this determines the propensity of the germination propensity. Okay, so if we apply this model, of course we can fit it. There are many parameters, there are many unknowns, so we could apply the von Neumann elephant idea that, of course, you have so many parameters that you can fit everything. The only thing that we can do is that we are not very gung-ho, we are not very obsessive at fitting the right parameters. We just want to see that for some parameters this thing works the way it does, and then we try to validate the model using other kinds of things, such as mutants. I'll come back to this in a minute. So let me already tell you that we are not very worried about getting the parameters. This is not a way of modeling, of measuring parameters, biophysical parameters. It's not that I don't care, it's that I cannot do it, so okay? But anyway, what we get is that the model fits very well if we find the right parameters, and of course, it reproduces well the thing. But of course, by itself, it's the von Neumann 5-parameter fitting thing. So the only thing that we can do is validate. And we validate by another thing that is very nice about bacteria is that you can have mutants. And one thing that Gurul has in his lab is thousands of different kinds of mutants and reporters of Bacillus aureus. Okay? So we can look at mutants in which basically the cell cannot import potassium when it's sporulating, and this alters the initial potassium concentration. And then when you have this mutant, then we see that the germination probability increases because spores have less potassium to begin with. And we can predict, of course we do the model, I'm not showing you this, but the model explains this. Okay, then we have two mutants that perturb the ability of the ion channel to export. One of them is just simply eliminating the ion channel. But this is also altering how much potassium I have at the beginning, so it's a bit misleading, but still, you can get some kind of results that you can explain with the model. And again, I'm not showing you this because I want to move on. And also you can block it with quinine, as I told you earlier, and then you see that there is very little sporulation, germination again. So, and the model reproduces all these mutants, okay? Again, it might be, we are just testing the model. And one more thing I wanted to tell you about this system is that, of course, there is a prediction. Maybe I can go back here. Of course, the model contains another elephant in the, sorry, it's the third time the elephant appears in this talk. But another elephant in the room is that one of the variables of the Hodgkin-Huxley model is the membrane potential of the cell. And I haven't said anything about the membrane potential. The question is, okay, what is the prediction? Can we measure the membrane potential? Is the membrane potential behaving the way we expect? And let me just tell you that the prediction is that the potential, we can call it electrochemical potential, is going to go up as so the prediction is that, this is a cartoon, no? As the germinal pulses appear, then the cell becomes, it increases the electrochemical potential because a positive charge is leaving the cell. So the cell is becoming more and more negative, right? So it's losing chemical charge. So it's becoming more and more negative. It's becoming more and more hyperpolarized, right? So that's and then so it releases potassium, it goes up. It releases potassium, it goes up, and this is predicted by the model, as you will see. But what I wanted to tell you is that we can measure the membrane potential using an Ernstian indicator, an Ernstian dye, and in our hands at least, theoflavin T, which is used for other things in life sciences, not to look for amyloid beta, no. But in our hands, we have over the years. We have interpreted that this fluorescent dye is an Ernstian indicator because it's the cell becomes more and more negative, and this dye is positively charged, so it's going to be the theoflavin T is going to be sticking more and more to the bacterial coat, to the spore coat. So this is a measurement of the membrane potential. So now let me show you here what the model. This is coming out of the model. It looks very straight, is because there's no noise in the model, so it's a bit sad, but that's the way it is. We could include noise, but it's okay. We don't need it, I think, because the important thing is that we see the electrochemical potential going up, the spore becoming more and more negative, and this is what experimentally we see here: different spores that germinate at different points, but all of them are following these steps, and when they germinate, they germinate. And since this is not, it's always nice to show a movie. This is a movie of the spores, of 4 different spores that germinate in response to different pulses at different orders, the first, the second, the third, and you can see how the membrane potential is going up. Okay? So anyway, this is what I wanted to tell you of this first part, in which basically the richness of the environment is temporal driving, periodic temporal driving of my environment, and which seems that our spores is using a mechanism that is very well known. I was telling you about Hodgkin-Huxley and neuronal dynamics. Of course, a very simple model of neuronal dynamics is the integrate and fire model, and what we believe is that these things behave in an integrate and fire way. You integrate the membrane potential, and then you get to a threshold and you fire. The difference is that here the fire is irreversible. In a neuron, the firing spikes. But here, the spore fires, it's gone, okay? Anyway, this is the first thing. So now, let's go to, oh, yes, sorry. Yes, please.
And in this case, obviously the effects of the strength of the pulse, the spacing of the pulse.
Yes, yes. So, yes, so for one, that's a very important, we haven't systematically done it, that's why I haven't, but you can, for instance, you could, instead of making the pulses every 2 hours, you could increase it. So we have increased it to 10 hours or to 20 hours, and we see that eventually the memory decays, because as you were seeing, the potassium is recovered because the spore take up the potassium, okay, that's it. So it's recovered in a few hours. So if you do it every 20 hours or even 10 hours, then this is lost. So but this is, again, if we interpret this in terms of sensitization, I mean, I don't know, I'm not aware of hallmarks of sensitization, but these things have been studied in habituation. There are dependencies on the frequency. So this is something that we haven't done. This has not been done systematically, but yes, we have analyzed this, yes.
And one more thing, aligns with the same question, the density of the spores, because it seems that the first one, if you have just one single spore, pulse doesn't tell them.
You mean density of the spores?
Yeah, the number of spores. But there is an effect from population.
Oh, that is a wonderful question. I don't think we have found this, but I may have been misleading, because the first slide that I was showing you, in which I was showing you one spore, this was just a crop. So this was not an isolated spore. So, I mean, here when I was showing you, this is a statistical measurement. You see everywhere. I don't have the data here, but we have the data in the supplementary of that paper. You look at the distribution of spores that respond the first pulse, the second, and there's no pattern. So they are homogeneously distributed independent on where they are with respect to the flow and with respect to the local density. But it's a very good question. We are assuming that these spores don't talk with each other. That's, but yeah, thank you. Okay, good. So now, this first rich environment was temporal driving, again. Now the second one is, again, also very simple, but completely different. So far, this is what we have been doing, a single signal that is temporal, but it's very simple temporal driving. But now I would like to discuss the city going from temporal complexity or richness to content complexity of richness, meaning that now I am going to consider, I mean, we could do the two things, but right now we cannot do it. We cannot vary both the temporal thing and the content. So what I'm going to tell you now is what happens if we have several, in fact only two, two signals simultaneously acting. They are more or less constant, so it's a problem of integration. So, which is very standard. So I'm pretty sure that you might be thinking, yeah, but it's true, it's very standard. But I wanted to tell you a story. This is an excuse to tell you about a story of signal integration by these spores, okay, because I'm continuing with these spores, which are very simple. So then the new thing is that these guys are super simple. So how do they do integration? So of course, I mean, cells can integrate all kinds of things, but I'm telling you about a little thing that is half dead. And you will see now the integration, okay? So two signals, more or less constant in time. We don't care about this. We are not imposing any kind of time dependence. And let me tell you, so what are the signals? So first of all, the first one you have already seen, it's L-alanine, okay? L-alanine, when you add it to a dormant spore, you're going to get germination, and that's what you see. That's so far so good. Now, interestingly, it is also known from the literature, not by us, that, and I don't know how this came about, but if instead of L-alanine, you use its chiral mirror, D-alanine, okay? If you use D-alanine, the opposite happens, I mean, nothing happens. In fact, D-alanine inhibits germination. And that was already interesting because, first of all, these are two enantiomers. These are two, so the only change between these molecules is the 3D structure, the chirality, okay? And they have opposite effects on the propensity of the cells to germinate. That was interesting. So then the question that we ask is what happens when you apply both. Let's analyze systematically how spores integrate these two signals. Okay, this is the richness right now, and this is done by another amazing postdoc in this case of Grohl, which is Colin Commercy. Yes. Because, well, we should look at the original papers. It's just that when you try to force them to germinate, you see less germination than you expected. So these things have been studied. So that's a wonderful question, but it's not no effect, it's inhibition. And in fact, so this was known, but I'm going to show you now the data. Okay, so that's okay. So I'm going to show you the data. So this is Colin's work. He did an amazing amount of work also. And I wanted to show you to you. So this is an excuse. Talking about rich driving or whatever, it's an excuse because I wanted to show you this story. So anyway, first, this is a first, well, it's not the first experiment that he did, but at some point we did it because it was a nice way of visually showing what was going on. Imagine that you have a large pad. This is an agarose pad. It's a very large agarose pad. And here you put a source of L-alanine, only here. And then this means that you're going to have a gradient of L-alanine. And then what you can see in this padding, which is full of spores, this is not a macrofluidics, we are not flowing anything, it's just static, we're just having the source of L-alanine. And then what you see is that as we expect, when green, sorry, I haven't shown you this, but you see it here. Green means germinated and magenta means dormant, okay? And what we can see is that as we get farther away from the source of L-alanine, we get less and less germination and we more and more keep going. So this is so far so good. That's nothing new. If you will, there is a little bit of a dose response analysis, especially. And the nice thing is that if we do now one in which we put the two, L-alanine on one side and D-alanine on the other, this is an amazing experiment that Colin did. I can say because it's not mine and my lab, I can say it. So I'm saying it, I think it's very nice. And you have here the source, and then you have the other source, and you see that indeed here you are near the D-alanine source, you really you have in a gradient kind of way really inhibiting the germination. So this is already showing indirectly. But on top of that, what I can show you is something like this. So now this is a movie in which you can see, well, these are 25 different experiments, okay? And this is a summary of 25 different experiments in which you have a bunch, you see a field of spores, and you subject them to different concentrations of L-alanine and D-alanine. And the movie is just showing that eventually, in time, well, there's a gradient of timing, but I'm not entering into the gradient of timing. That's another issue. There's a gradient in the response time, but I'm just looking at what happens at the end. And what happens at the end, you see that this is already stabilized, okay, after 1 minute and a half or something. And what happens at the end of the experiment is that you have something like this. So if we can calculate the germination rate, in fact, we are not looking at the percentage of spores that have germinated. We are looking at the speed at which the percentage of germination changes. That's what we call the germination rate, percentage of cells germinated per minute. And we see a nice behavior like this. This is our integration map. So we can call this an enantiomer integration map. This is the way that this thing is integrated into things. And we go back again, and this is showing you that as we go up in the alanine, we get less and less germination. So this is another way of showing that we are inhibiting, okay? But we are inhibiting in a very special way. We are inhibiting in a ratiometric manner. So what this is detecting is the ratio. I mean, the fact that we have a line here, it's also very nice. We have a line almost. I mean, we could fit a line. And if we fit a line, this means that everything depends on the ratio of the two enantiomers. Yes?
Sorry, so you put the alanine just in this, you apply it just in this up here?
Sorry?
So there's a square. And
Yes.
you only apply the alanine on this, on the bottom,
sorry, there is no, I'm not sure if I understand. So there is no gradient here. This is just a path in which you add L and the alanine. There's no microfluidics, there's no gradient. So everything is homogeneous, but I think this is not what you mean. I apologize. But if you, for instance, here you have in this area, you have a very small concentration of L-alanine, and you increase the concentration of the alanine. That's all I'm saying. So here, since the concentration of L-alanine is so small, nobody germinates. Okay. But if I go here, maybe let's do this one here. This concentration of L-alanine here. And again, you can see, going back to Robert's question about the concentration, we are talking about much smaller concentrations because this is not a pulse anymore. It's just a total, it's constant germinant because I'm not. So that's why now we are talking about 0.1. So for instance, in this one, you have a reasonable concentration of alanine that induces germination, but if you have a lot of D-alanine, there's nothing happening.
Oh, okay.
Okay, so it is that really an integration map.
But wouldn't you see the same results if you just don't apply the alanine?
If I don't apply, so sorry, let's take a look at this direction. So for instance, if I don't apply the alanine, I'm here. And we can define a threshold of L-alanine for which I have germination on this side, because here there's nothing, there is not enough L-alanine to produce germination, and here you already have some germination. And this is, let me remind you that this is basically the steady state. This is saturated, as you have seen in the previous movie. So as you are seeing here, nothing changes anymore after a while, or hopefully, I don't know. Yeah, well, maybe it does. It doesn't really matter right now. But what I mean is that you can see that if you have a low concentration of L-alanine, there's no germination at all. And at this level, you already have some germination. So when you increase, you find that the concentration or the threshold, no? What I'm saying is that as you apply more and more of the alanine, the threshold concentration becomes larger. You see that so what we are seeing here is that the threshold, so for a fixed D-alanine, the threshold, you need more and more and more and more L-alanine to produce germination. And you do it in a linear manner. So it's a ratiometric response. And the ratiometric responses in integration of things have been studied for many years in many different situations. My question here is that what is causing this ratiometric integration when what I have here are rocks? These spores are like rocks, stones. It's like, they're almost not alive. And yet they're able to integrate and generate a ratiometric response. Why is that? So I'm already too late, but anyway. So our assumption is very simple. The only thing that you need is competitive binding of the two enantiomers to the germinant, which I guess you were assuming, because of course, if the only thing that changes is the chirality, you can assume more probably the two germinants are going to bind to the same receptor competitively. The only thing is that the D version is not going to activate, and this we have no idea why, but this is the way. This is an assumption that we are saying, look, the two of them bind competitively. This is the simplest assumption that I can imagine. And then only the D-alanine is not activating. If you do this, you can very simply come to an expression for a germination rate in this very simple model in a steady state. This is a steady state model in which the germination rate has something like this. This is competitive binding on L and D to the germinant, receptor. And then this is, let me remind you that this is an experiment, and which if you plot this thing in 2 dimensions, there's nothing else to it. If I plot this thing in 2 dimensions, this alpha is basically telling me about this height, but these two things, KL and G, are basically, you can use to fit this. And what you get is something like this. Maybe it's not so, we did it in this is very scarce, but here you can do it with infinite with infinite precision, well, very large precision. And then you see that this thing grows very quickly from 0 to 8, more or less, percent of germinated spores per minute. So we get this ratiometric response. Okay. So, well, there is only one thing to tell you, that of course here I only see this line, and here the model predicts that this thing is dropping. No, I'm not. You can see it. So this like this. I haven't drawn a contour. I could have plotted a contour line here at, I don't know, at whatever threshold, and you see that this is doing something like this. So these levels off at low D-alanine levels that we do not see experimentally, but this is something that I will show you in a minute why we have it like this. Okay. Anyway, sorry, why is this? So now we could try to ask the question of why. Why is the cell, why are the spores doing this kind of integration? Why? And then here we come, this is another speculative thought. This is our speculation, okay? We believe it's probable. So anyway, so I have to tell you, L-alanine is an indication that there are nutrients. L-alanine is produced by all forms of life, produced and consumed, because most amino acids, you know better than I, are levo. So the chirality of the amino acids, more common one is levo. And in particular, L-alanine is the case. So cells of all kinds are exchanging and producing, synthesizing and consuming L-alanine, and this includes all kinds of life forms. And this includes eukaryotic life forms, plants. If we are, Bacillus satilis is in a soil bacterium that sometimes maybe coexists with plants. So if there are plant cells, there are going to be L-alanine. So for the spores, they interpret L-alanine that they're going to be nutrients, so I should wake up. But what about D-alanine? D-alanine is very curious, because you might be thinking, why is this useful if most of the amino acids are L? Well, it so happens that bacteria need D-alanine to build their cell walls, okay? To build those cell walls, they need D-alanine. And this means that they have to produce it. So this means that bacteria release D-alanine, but not eukaryotic cells, not plant cells, not whatever. So this is our assumption is that the spores are using D-alanine as an indication that there is other bacteria. And if there is other bacteria, I have to tell you, when these cells are born, these are like little babies. They're very weak. So if they are born in a field of other bacteria that are consuming nutrients like crazy, these guys, so our assumption is that it's not good for spores to germinate when there is a lot of bacteria around. So these spores need a mechanism that we could call an anti-quorum sensing, in the sense that they need to know when there is people around, bacteria around, because if there are bacteria around, I am not going to germinate, okay? So is it true? Well, let's see. So now what Colin did, the next thing he did is, okay, I'm gonna take a bunch of bacteria, species, these ones, okay, 20 or something. I'm going to culture them, I'm going to eliminate them, and then I'm going to take the supernatant and I'm going to put this through a mass spectrometer to see how much L-alanine and D-alanine these guys are producing in normal conditions, in vegetative conditions, in classic laboratory conditions. Okay, and what he saw is that this, and this is pretty amazing because this comes after well, to be honest, we use this because we saw this. You see all the, of course, there's a lot of variability, but they seem to be around, outside the region where the bacillus spores are germinating. And this is like this, if you will. Of course, there are not so many, but here they are on this area. Okay, so to be honest with you, we also use this fact to really fit this because we could have moved this, but the fact of the matter is that this integration function has this shape no matter what. And it's we can fit the parameters to fit this slope and this position. But the fact of the matter is that this thing is obeying this kind of thing like this. So it's very interesting. So is it really the case? So what happens if so maybe the next thing is, what happens with the spore? So can I test this? Can I just co-culture? Let's say, or sorry, not co-culture. I'm going to take the media produced by these cells and put it on the spores. And then when you do it, you see this is different now because what this is telling you is this color is telling you, look, this particular spore, this particular bacterial species is producing this type of L-alanine and D-alanine, and the color is telling you whether the spores are going to stay dormant or germinate. Okay, so this is just taking the medium produced by the alive cells and applying them to the bacteria, And what we see is that everybody that is outside this region is not germinating, is not inducing germination in bacillus. And only this cell, in particular this one, which is well inside this region, the L-alanine and the alanine that they're producing, the ratio, which is what we are measuring, is basically enough to permit Bacillus to respond. But the other ones, they don't. So it's as if Bacillus has evolved the binding, the dissociation constant between the two the germinants and the receptor to avoid germinating in the presence of all these other bacteria. Maybe not these ones for reasons that we don't know. Okay, this one is not a threat. But the other ones are a threat, I'm not germinating, okay? And the final thing is that, well, we can do co-culture. This is not co-culture, but we can do co-culture experiments. So another thing that Colin did is, now let's put together the spores with other species of bacteria. Let's force the spores. Now I add a lot of L-alanine, I force the sporulation even if the bacteria don't want to, and then we see who survives. If the spores die, then it's clear that these spores are not in a competitive, I mean, it's not advantageous for them to survive in the presence of this bacteria. If the species is such that the spores survive and grow happily, then it's better to be germinating. And when he did the entire analysis for all these species, what he got was this, that for most of the spores, they are better, and so the bacillus is better if it stays dormant. That's what I'm saying. It's better for bacillus to stay dormant if they are being co-cultured with other bacteria that are alive producing the alanine. Okay, so I'm telling you, it's as if there is a social avoidance mechanism. So this thing, I mean, look, spores that have no energy, they are able to implement a social avoidance mechanism, an antiquorum sensing mechanism, not to do something if there are other bacteria around. This is the conclusion.